In this module, we will talk about the range of fermentation product, especially which will come the third range of fermentation process scope, that is enzyme production. So we can produce a number of enzymes through fermentation process. As we know that the fermentation process is the mass culturing of the organism for the sake of desired product. When our focus is the enzymes as a product, we know that when organisms grow, enzymes are the essential part of the enzyme of the cell. We can say that the enzymes are the fountain of life. So different metabolic pathways are governed or catalyzed by different enzymes. So if we run our fermentation process to produce the enzymes, so enzymes, as we say, they are the functional proteins with few exceptions. So in particular, when we talk about the enzyme with respect to fermentation process, enzymes can be classified into two categories: extracellular enzymes as well as intracellular enzymes. As concerned the intracellular enzymes, these are those which remains inside the cell or the cell organelles and responsible catalyzing all those processes which need the cell. But on the other hand, the extracellular are those enzymes which are released by the cell to perform a job external to the cell. There are so many enzymes, as we can know, that digestive enzymes. So on the other hand, if we talk about the enzyme with respect to fermentation, enzyme can be classified into two further categories. One is constitutive enzymes and other are inducible enzymes. So what are constitutive enzymes? So these are those enzymes that is synthesized continuously regardless of the growth conditions. Whatsoever the growth condition during the fermentation process, these constitutive enzymes has to produce in every cost. But on the other hand, the inducible enzymes are only those enzymes which synthesize only in the presence of substrate that act as inducer. We all know that enzymes are the functional proteins and they are produced in result of central dogma, that how the gene express and then how they formulate and then they express a protein, then there is a functional protein. So if we design a fermentation process in order to produce the inducible enzyme, we have to put some inducers in the medium which act or suppress or in a stress activate the cell to produce such enzymes. There are a number of examples of extracellular enzymes, intracellular enzymes, inducible enzyme and constitutive enzymes. Here you can see that one example of inducive or inductive enzymes, which you call as serves as pyruvate carboxylase, that enzyme which turns pyruvate into oxaloacetate, which is synthesized on demand and the process is activated by cortisol. As an example of extracellular enzymes, we know that there are cellulases. Especially we talk about when we grow some filamentous fungi in a fermentation process. Mostly the fungi require monosaccharide for their growth as a carbon source, but when the monosaccharide or disaccharide not present in the medium and then there is a polysaccharide, cellulose is present, then that microorganism require the glucose or you can say the xylose as a carbon sugar. So the presence of cellulose act as the inducer on such filamentous fungi that enable that fungi to express his cellulase system, and then the cellulase group of enzyme produce and then excreted outside the cell, and then when the cellulase is excreted out from the cell or remain inside the cell, they catalyze the reaction of conversion of cellulose into glucose, and then glucose is utilized by the organism to grow and to regulate different metabolic pathways for the need of energy. So, during this fermentation process, we can use the fermentation process of a number of enzymes. We know that in a single cell, number of thousands of enzymes are reported in a single cell. So, by this fermentation process, we can use this process for the production of number of enzymes.